Good morning, students. Today's topic of discussion is chi-square test. Now, chi-square test is a very important test in the sampling distribution. Now, chi-square test was developed or propounded by Pearson, and this was done way long back in order to measure the various frequencies and compare data with whatever was required. Now, this test compares measurements relating to the frequency of individuals in defined categories. For example, the number of white and purple flowers in a population of pea plants. Chi-square is used to test if the observed frequency fit the frequency you expected or predicted. Now, how do we actually calculate the expected frequency? We may predict a homogeneous distribution of individuals in an environment, and we can take the expected frequency. Now, I'll be discussing null hypothesis. What is actually null hypothesis? It is defined by H0, and there is no statistically significant difference between the observed frequency of maize grains and the expected frequency. So, if there is no statistical significant difference, that is taken as a null hypothesis, and the alternative hypothesis is taken as significant difference between the observed frequency of maize grains and the expected frequency. Now, if the value of chi-square exceeds the critical value, that is p equal to 0.05, then we can reject the null hypothesis. And if the value of chi-square do not exceed the critical value, we can accept the null hypothesis. Now, how is this measured? Chi-square is given by summation of O minus E whole square by E, where O is the observed results and E the expected results. Now, whenever we take into account this chi-square, we have to find out the degrees of freedom. The degrees of freedom means number of categories minus 1. Now, if we calculate the chi-square for a particular set of information, and we see that there is a significant difference between our expected and observed ratios, then we consider that it is a poor fit, it is not a good fit. Now, we can take another example also, like observed frequency of fish species and the expected frequency, and we can calculate the similar chi-square and get the results. That is how we actually take the results into consideration. Now, what is a contingency table? A contingency table is used to calculate expected frequencies when the relationship between the two quantities is being investigated. Now, these contingency tables are important since they form an important part in the determination of chi-square test and observing them. Thank you. Have a nice day.